phylogenetics and microbiology. He was then a research scientist at Stony Brook and a research assistant professor at Stony Brook, and joined us here at Rutgers in 2017. And he's going to be talking about some of the research that he has done since.
Yeah.
Right. Thank you.
Thank you for the introduction, Dina. So I hope people on Zoom can hear me well. If not, please let me know. Today I'm going to be talking about the invasive longhorn tick, Amaphysalis longicornis, and what we have done in terms of studying its phenology and control. But before I dive into today's talk, I want to summarize part of other work that I do in my lab that I'm not going to be presenting today. And for that, I'm going to use the triad of vector-borne diseases that I'm showing here. The pathogen that I study is Borrelia burgdorferi, which is the agent of Lyme disease. This pathogen is vectored by Ixodes scapularis, and although I'm not going to talk much today about Ixodes scapularis, we have done also a little bit of work with it. And it bites sometimes humans, producing Lyme disease, although their typical hosts are rodents and other wild animals. Now, the environment is critical for these interactions because it modulates the habitat of the tick, but also the density of these rodent populations. So in my lab, we have or we are studying different aspects of this triad. For example, in terms of Borrelia burgdorferi, we are interested in studying the role of cholesterol and fatty acids in membrane order. We are also interested in studying certain proteins, particularly those involved in moonlighting. And this is quite interesting because we know very little about moonlight proteins. These are proteins that are found in atypical subcellular localizations. And one example of them is GrowEL, that an undergrad student did a project in my lab and actually ended up publishing on this. Is a chaperone protein present in the cytoplasm, but it's also found associated with the membrane. And we saw that this chaperone is actually involved in binding extracellular matrix components and also plasminogen. And extracellular matrix components are probably critical for the bacteria to establish the infection. And plasminogen is the inactive form of plasmin, which is a protease found in the body of mammalian hosts. Borrelia doesn't have exoproteolytic activity, so it borrows it from the host. I also working in the context of Lyme disease in hypercholesterolemia in the host, and this is a manuscript that actually we are preparing right now. Long story short, we have shown that hypercholesterolemia in the host enhance the dissemination of Borrelia spirochetes early in the infection, and also the burden of these spirochetes is much higher in mouse with high cholesterol. We are also working with ticks. Jen Mora is working on developing an in vitro feeding system with the aim to study acaricides and repellents in a more in vivo-like setting, if you will. We are also have a project in detecting tick pathogens in rescue wild animals. And today I'm going to be talking about specifically about longicornis, a little bit of phenology and control, which is most of the work that done by either Julia Gonzalez or Matt Bickerton. And I'm not going to present everything that they did, but just a flavor of it. So let me start today's talk with this outline. I'm going to give a brief introduction on Amaphysalis longicornis. I describe it a full hiding in the open because when we first detected Amaphysalis longicornis, it was already present here for quite some time. We don't know for how long, but clearly present from several seasons before at least. Then I'm going to go a little bit on the phenology of ALT, and then on some work that we have done in terms of control and prevention. So the Asian longhorn tick was first discovered here in New Jersey, and here's the short communication by Thor Rainey, Gene Ossie, and Andrea Gysi. The three of them intimately related to the Center for Vector Biology. Thor Rainey was sent to a farm and he to treat a sheep that was infested, and the area surrounded the sheep. And basically, he couldn't identify morphologically the tick. Got in contact with Jim Olsey, with Andrea Gysi. She ran a CO1 barcoding, and it came out 99.9 similar or identical to Amaphysalis longicornis. They then did some morphological studies and published this first description of this invasive tick species in the United States. And this is what it triggers all the research that came afterwards. The ALT is a tick that is important from a medical and veterinary standpoint. In Asia, this tick species can vector severe fever with a thrombocytopenia virus. It can also vector Rickettsia japonica, which is the agent of Japanese spot fever. And these are, of course, human diseases. In the United States, there have been some studies looking at the vectorial competence of this tick species in the transmission of several pathogens, including Borrelia burgdorferi. Is not a vector of Borrelia burgdorferi, but is a vector, at least under laboratory conditions, of Rickettsia rickettsii, which is the agent of Rocky Mountain spot fever and is deadly disease if it's not treated. But ALT is also an important cattle ectoparasite. It produces direct damage by decreasing the production of cattle, but it also is involved in the transmission of pathogens like Teleria orientalis. And this pathogen has been linked to cattle deaths in Virginia. So we know this tick has the potential in the United States to be involved in death of cattle through the transmission of a parasite. So the Asian longhorn tick is a three-host tick species. For those of you that are not that familiar with ticks, three-host tick species are tick species that feed on a different animal each time. They feed, drop off, look for another animal, feed, drop off, and they do that as an adult before they lay eggs on the soil too. It has a broad range of pests, so it's not extremely specific, but contrary to other tick species that are a problem here in the transmission of disease to humans, like Isodes scapularis or Amblyoma americanum, they don't really seem to be using rodents that much, and they are not extremely anthropophilic, meaning that they don't typically bite people, although this is always the result of not only their anthropophilic behavior, but their numbers. There are two types of populations in Asia of ALT. They have bisexual populations and parthenogenetic populations, and these populations don't mix together. They are separate. This is something kind of, the presence of parthenogenetic populations is kind of unique for this tick species. You don't find it in ticks that much, but you find it a lot in mites. And in mites, in fact, you find bisexual populations or populations that can reproduce sexually and asexually at the same time. The bisexual populations are in Asia, but the parthenogenetic populations, which are the invasive populations, are present also in Australia, New Zealand, and now in the United States. One of the characteristics of these populations is that they can reach high numbers from single individuals, and that makes them very invasive. And here you have a very nice picture from Gene Ossie taken from the field, and this is not uncommon to see large number of ticks clustering together. Clustering in ticks is very, very rare because you typically see them a few of them, but in the field. But it's really hard to find any type of clustering. There are not that many. but that's different for these tick species that can reach very high numbers. Now, there were a couple of predictive models showing areas that with high habitat suitability for Amorphosalis longicornis in the United States. These models were based on known distributions of these species in Asia, New Zealand, and Australia. And as you can see, great part of the United States is a highly suitable area for Amorphosalis longicornis. And in fact, if you look at these predictive models and where we have found these tick species so far, it kind of mimics quite well what it was predicted in these models, particularly by Ilya Rotzlin too. Right now, this tick species has been detected in 19 different states and over 220 counties in those states. And now I'm just going to start talking more in depth about the work that we have done, starting with the phenology of ALT. And these are two phenology studies that Matt Bickerton conducted, and in which we look at the presence of these tick species over time. And what we realize is that this tick species starts really early on. Around the first week of March, they are already out, and they last until basically the last week of October. And this is kind of this happens every year. We have done this multiple times, and we know now when to expect ticks out and for how long they are going to be out. The phenology follows kind of a logical fashion in sense that you find nymphs first, then adults, and then larvae. This doesn't happen with all tick species. Sometimes you see, for example, in Ixodes scapularis, you first see nymphs, then a larva, and then adults, and that has to do with how they overwinter. But for this tick species, it's very straightforward. Now, I want just to mention that the number of larvae is much higher than the number of nymphs or adults. That the axis for larvae and the numbers are different from adults and nymphs. So we have a lot of larvae by default. But another thing that we saw in here that called our attention is that there's a lot of overlapping that we don't typically see with other tick species. We see, and it's hard to see here, but we see a larva in the early spring. We see nymphs like really late in the summer, and sometimes almost in the fall. And you might think that these are very, very low numbers, but they are not that low when you look at the proportion of ticks, because these axes are different. So you might have 25% of your ticks collected in May and June might be actually larvae. And that basically suggests us that probably there were more than one population of ticks. That larva was probably comprised of populations that were different and had different origin. And that is important if you want to control the tick in terms of developing plans for controlling. I'm not going to talk about this specific project right now, but we look into the different cohorts of tick by looking at the energy budget. And the energy budget we measure as the lipid content that these ticks have. And by looking at this, we can determine for how long they have been on the field. Remember that ticks only feed once per stage, so the energy budget that they have initially is going to be much higher than the energy budget that they are going to have by the end of the life stage before they feed. So Matt will be presenting more about this energy budget project in his defense, which is within a month. So good luck explaining that part, Matt. I'm gonna now jump a little bit on a small project that we did in collaborations with Dina. We look into the presence of these different tick species in the ecotone. We were interested in the ecotone because the ecotone is the transitional area between the forest and the path that you find in parks, and is typically the area where people are more exposed to ticks. People and pets inadvertently can go into the ecotone more often than they actually do deep into the forest. So we thought that by looking at this area, we can assess what tick species were more problematic and people were most exposed to. And this is a similar graphic showing the different tick species that we found. We found Amphiphysalis longicornis, Ixodes scapularis, which is the black-leg tick, Amblyoma americanum, the lone star tick, and Dermacentor variabilis, which is the American dog tick. We find all stages for all species except for Dermacentor variabilis. This is not a surprise because Dermacentor variabilis, the immature stages are endophilic, so they don't typically quest and are hard to see. Most of the individuals that we collect were Amphiphysalis longicornis, so we found large number of Amphiphysalis longicornis, decent numbers of Ixodes scapularis, and also high numbers of Amblyoma americanum. The ecotone is kind of a harsh environment for tick species that require high moisture, which is the case of Ixodes scapularis. One of the issues that ticks have is that they cannot regulate their water content very well. So in areas where, like the ecotone, that you don't have as much moisture as the forest because there is less left litter and they are more exposed to the sun, species like scapularis suffer more. So one of the things that we did in parallel, besides looking at the distribution of the different stages throughout the year, was to look if the environmental conditions play a role in the abundance of questing ticks. And we found a positive correlation between temperature and saturation deficit, which is saturation deficit is a way of measuring temperature and relative humidity together. A positive correlation of both of these variables with the presence of adults of longicornis and adults of Ixodes scapularis. For Ixodes scapularis, it's not a big surprise because Ixodes scapularis adults are present in the fall. Just by experience, we know that when we go out in a warm day in the fall, we kind of see more ticks than if we go into a cold day. So this correlation kind of makes sense with our previous experience. Now, what it really was striking was the presence of higher presence of adults of longicornis in warmer days or hotter days in the summer, because these ticks are the adult stage of longicornis is present in the summer, and the summer is typically hot, and this data suggests that it's a tick species that is very well adapted to hot environments and drier environments. So much hotter and drier than we find in New Jersey, which makes them actually very adaptable to colonize transitional areas in areas like New Jersey. So the conclusions for this study was that the ecotone of parks and recreational areas in New Jersey is a suitable habitat for important tick vectors like longicornis, black-leg ticks, and lone star ticks. That the Asian longhorn tick is well adapted to the ecotone and transitional areas, and the abundance of ALT and black-leg ticks adults correlates with a higher temperature and saturation deficit, which is might be a surprise for ALT, not that much for black-leg ticks. And then something that we saw is that the proportion of different stages was similar for longicornis and Amblyoma americanum, which are two species that are well adapted to hotter and drier environments compared to Ixodes scapularis. And that proportion was skewed for Ixodes scapularis. They have a 5% of nymphs and a 10% of adults. And this can mean two things. One is that they might exploit different type of hosts that move them around differently. But the second part of this is that this might be a harsh environment for Ixodes scapularis ticks, particularly those that are exposed there when they overwinter. Now, another thing that we did in terms of phenology of ALT was to look into the effects of photoperiod in development. We look into larvae and nymphs particularly engorged larvae and nymphs, because if you remember the figure that I presented before, larvae are present in the fall. That larvae are going to feed, are going to become engorged larvae. They're going to molt into nymphs, and nymph is the primary stage that overwinters for the Asian longhorn tick. So we wanted to see whether engorged larvae will, their development will be affected by the photoperiod. So we compare engorged larvae and engorged nymphs under short day length and long day length, right? One mimicking more conditions that happen in the fall, early winter versus conditions that happen in the summer. And we look at the percentage of molting and the mean of molting time, the time that takes them to molt. And we saw that there were no difference when we look into engorged nymphs, so it doesn't seem to be affected by the photoperiod, which it makes sense because they are not in a hurry to molting really into the next stage. But that seems to be different for engorged larvae. Engorged larvae, they successfully molt, or they molt better in short day length, and it takes then less time to molt in short day length than long day lengths. And this probably is a cue that engorged larvae use that allows them to molt faster before overwintering, because we suspect that overwintering as nymphs is much beneficial for them than overwintering as engorged larvae. So, and one thing that I mentioned before is that ALT is not particularly anthropophilic, and this is true. But there have been instances of people bitten by these tick species. If you remember, the parthenogenetic populations reach high numbers in small areas, and when you have high numbers in small areas, that increases the risk of tick bites. It's like playing the roulette. You have a small chance to get the number, but if you play enough times, you're going to hit the number. So that's what happens when you enter an area in which the density is very high, that your probabilities of getting bitten, even if the species is not particularly anthropophilic, increases. Now, patients that get bitten by Asian longhorn ticks develop an erythematous pruritic lesion, which is just refers to a lesion with a small redness, slightly elevated, and that is itchy. That resolves itself typically in 2 weeks. Since this is an invasive species, it recently came here. It's not clear whether they can transmit human pathogens yet, at least in nature. We still think that public health practitioners should become more familiar with this tick species, and we need to complete probably more bacterial competency studies to determine what pathogens actually can be transmitted by this tick species. Now I'm gonna move a little bit to control and prevention, and I'm gonna start talking about. We're gonna start this part talking about catnip. Catnip is a plant, Nepetaria catania. This project was done in conjunction with Gene Simon from Plant Science. He developed 2 cultivars that were enriched in nepectalactone, and nepectalactone is the active ingredient involved in the repellency from this plant. He also makes some modifications to the plant so it can be actually grow in big farms and be collected for production. So we look into the repellent properties of the 2 extracts of these novel cultivars in Amaphysalis longicornis and in Ixodes scapularis. And we look into, we use 2 extracts that Gene Simon provided to us, CR3 and CR9. They are actually very similar. They have very minor differences, and both seems to repel ticks quite well, at least in vitro experiments in the lab. we saw a small difference that initially we didn't know what to make out of it between Longicornis and Scapularis. It seems that it repels Scapularis better than Longicornis, but with this type of experiments, it's tricky to make any conclusions looking at these comparisons. We also were interested in comparing these extracts to repellents that are already used in the market, like DID. So we used 10% DID, CR3.9 and CR3.3, because DID 10% is typically the minimum that you find on repellent products. And we didn't see any differences when we compare the three of them, at least in these in vitro experiments. Not for Haemophilus longicornis, neither for Ixodes scapularis. So one thing that is critical for repellents is to know for how long these repellents maintain or keep their activity. These repellents are repellents because they are volatiles, but you have to be careful with this repellent. The more volatile they are, the less longer that their effect is going to last. So we wanted to see whether these have similar terms of control over time, which will suggest that we have similar properties in terms of volatility. And we did this with DID, CR3.9 and CR3.3 for Haemophilus longicornis and Ixodes scapularis, and we didn't really see major differences among them. They seem to behave similar. So all those things were good news. Then the next stage was we wanted to do is to look into what was the main organ associated with repellent for CR3 in Haemophilus longicornis and Ixodes scapularis. Ticks, unlike insects, they don't have antenna, but they have an olfactory organ called Haller's organ. Haller's organ is probably not only an olfactory organ, it does other things. It can also detect infrared light, for example, which might help ticks to quest and detect the host. But it's we know it's fundamentally the organ that they use for olfaction. So what we did in this experiment was to basically remove Haller's organ from Haemophilus longicornis and from Ixodes scapularis, and see what happened when we put them in the presence of CR3. And what we saw is that there was a decrease in repellency, but there was still higher repellency of these guys compared to the controls in both cases, which suggests that the bulk of this repellency is mediated by Haller's organ, although there might be additional sensory receptors involved in repellency. And we know that ticks have receptors in the palps and in other parts of their body that they might also play a role in repellency. So the next study that we did was to compare directly DID with CR3, because we have make all these comparisons indirectly. In this case, what we did basically was kind of create a two-choice experiment, put the two repellents and see what happened. And this study was very interesting because the difference were striking between longicornis and Ixodes scapularis. So when we put longicornis in a two-choice assay and we put CR3 and DID, this tick always go to the CR3 treated area. So DID overpowers the extract, at least for Haemophilus longicornis. When we did this same experiment with the scapularis, we found that for scapularis work it the other way around. When we put equal concentrations of CR3 and DID, basically the tick tend to go to the area treated with DID. And this was kind of surprising initially, and what we start doing is looking if this was a dose-dependent response, and we start lowering the concentration of CR3, and when we start lowering the concentration of CR3, we start seeing that then ticks were repelled by DID. So when we use 5%, then we didn't find significant differences in repellency between DID and CR3. with 1% clearly did repel much better, and with 0.1% it's like if we didn't have anything in there, it was all did. This was very interesting, so we decided to repeat the Haller's organ experiment, but now doing the two-choice assay. We again remove the, this is with the Haller's organ intact and with the Haller's organ removed. For Amaphysalis longicornis, well, when we remove the Haller's organ, then ticks didn't care whether to go to one place or to the other. There were no significant difference between them. But when we did the same experiment with the scapularis, when the Haller's organ is intact, they fundamentally go to the area treated with Dept. But when we remove it, they preferentially go to the area treated with CR3. So what we think is that the way Ixodes scapularis is detecting Dept and CR3 might be different from the way Amaphysalis longicornis is doing it. And Ixodes scapularis is a prostriata tick, is a primitive tick, quite different from all the others, which include longicornis, Amblioma americanum, the Marcentor variabilis, and so on and so forth. So there might be significant difference in how Ixodes scapularis and Amaphysalis longicornis detect these repellents, and we're interested in understanding that better and looking into maybe olfactory signatures in these ticks associated with repellency, and how that happens particularly in Ixodes scapularis. And to finish this part with catnip, I wanted to put this figure here because one thing that we observed when we were working with nepectalacton with the estrats is that ticks exposed to CR3 tended to die quite rapidly when we put them back in the incubator. I mean, we were testing this as a repellent, not as an acaricide, but since we started observing that, we just decided to conduct a study and look at what happened if we expose them, and we only expose them for 30 minutes. We expose them for 30 minutes, put them back in the incubator, and see what happened. And we see that they clearly are dying much faster than ticks exposed to Dept. So probably this is worth looking more into whether this has the potential to be used as an acaricidal too, because it seems to affect the survivorship of ticks. And now I'm going to jump to laboratory and field assays. That these studies were done primarily by Matt Bickerton. I'm not going to talk about all the work that he has done, but so I'm not going to talk anything about Boveria bassiana, for which we have some exciting data, but I'm going to summarize part of the work that Matt did for controlling longicornis in the field. And the first study that I'm going to show you here is the one that he recently published about the baseline susceptibility of ALT to acaricides. And the way we conduct this study is we have longicornis larva, and we put this larva in what is called larval packets. So these larva packets consist of Whatman paper that is treated with acaricides. And then we create this, we secure these Walmart papers making package with binder clips, and we put the ticks in this binder in these packets and leave them in an incubator with high relative humidity. And then we look into mortality. Matt figured out this dose response of estimates for carbamates, including propoxur and carbaryl, for tiotroids, including bifenthrin and permethrin, and for the organophosphate coumophos. We choose products that can be used for tick control or that have been used for tick control. Besides creating this these figures to figure out the LC50 for each product. One thing that was very interesting is that Matt compared all of these to data that was already published for other tick species, because we don't have a strain resistant to any type of acaricide that we can compare our population with, right? So if we cannot make those comparisons, but at least we can compare what we see in our population with other studies on ticks. Most of these studies are limited. I mean, these studies are limited in ticks, mostly done with Rhipicephalus microplus, which affects cattle, and with Rhipicephalus sanguineus that affects dogs. But what we found out when we compare the data from these studies to our data was that in red is the data that Matt generated, is that for all these products it doesn't seem to have resistance. It is on the low margin of values or within the range that you find in other studies. So it seems that ALT populations is not resistant to acaricides. And now I'm going to talk a little bit about some study work that Matt did. And this study works are extremely intense. I'm just going to present the results, but bear in mind that he did all the treatment. He counted all those ticks. He counted thousands and thousands and thousands of larvae, to the point that one day he came to my office and said, "I'm dreaming. I am counting larvae." So that's how intensive this study was. So the first one that he did, we wanted to figure out a way to control ticks in the field that didn't require continuous treatments over and over. So we decided to design a staged target acaricide application. We knew about the phenology of the tick, so we knew when to expect the tick, and we could target specific stages, and then see how that affected the population, and if we could achieve control. So we use lambda-cyhalothrin because it has good residual activity. It was proven to work for ALT in other studies, and it's registered for use on ticks in the environment. So we use lambda-cyhalothrin in this first study. And the idea was to basically target the peak of nymphs, the adult peak, or the end of the adult peak, beginning of opposition. So we did 3 types of treatments. Treatment in June to target the nymph population, target in July to target the adult population, or a treatment in August to target the end of the adult population before they had the ability more towards when they were about to lay eggs. And then we compare that with treating them in June, July, August, and June, July, and August. We did this in a field, in a park, and what we concluded from this study is that lambda-cyhalothrin was effective, which was expected. Now, foliar applications of lambda-cyhalothrin control 100% of ALT for 7 weeks in the environment. That happens in all studies, and we think that after those 7 weeks, what happens is that ticks are likely reintroduced from animals into that area again. June application provide control of the nymphal stage, but it had no effect later on the subsequent adult and larva population. The July application provide 70% control overall, but we still saw a little bit of larva coming out later in the fall. And the August population basically controlled 99.8% of ticks from August to October. So it did very well. And then the 3 applications provide 99.9% of control. So basically what Matt proved here is that with this type of control, you can achieve control very similar to treating 3 times a year just by treating in August in terms of controlling the population overall. And then he followed up this study using another one. In this case, now that we knew that targeting the population when they are about to lay eggs can kind of confer the better control, wanted to study different types of acaricides, and we also want to study whether insect growth regulators could provide control in the field. So insect growth regulators have been shown to some extent to work under laboratory conditions, but the truth is that ticks and insects are quite different, so I was a little bit skeptical if it would work in the field, but there was some preliminary data on in vitro work for Amblyomma americanum, for example, that shows some promise. So we decided to put it in here to use alternative to typical acaricides. So lambda-cyhalothrin and bifenthrin, they are both pyrethroids, but the formulation is different. Lambda-cyhalothrin we give it as a spray, bifenthrin as granules. Carbaryl we use as granule, it's a carbamate, and then the insect growth regulators. We did something similar. This is when we did the treatments in July and in August, the adult peak and the ovoposition. And in this case, we also wanted to know the impact of these approaches, these acaricides, on not target insects. And for that, Matt used sticky cards and pitfalls. The idea was to collect the data in non-treated areas versus treated areas, and then see what was the impact on them. So what we saw for the lambda-cyhalothrin is that in July 14 provide good control, a little bit better in August 5, but this was basically our positive control. We knew it already worked because we did it in our previous study compared to the untreated. Okay, then we look into bifenthrin. Bifenthrin, also a pyrethroid, provide also good control, not as great as lambda-cyhalothrin, but very close to it. So granules or liquids, they both sort of control almost equally well the tick population. Now, when we use carbaryl, to our surprise, carbaryl didn't do much. We only saw a 22% reduction in July 4th, which is almost nothing, and we didn't see any differences with the August 5th application. So carbaryl didn't work, and when we use pyrifloxacin and Pernoff, insect growth regulators, we didn't see any difference either in terms of control. So basically, only the granule and liquid pyrethroids were the one to kind of control. And here you can see this in summary of this by species, by stages, and by treatments, using the different acaricides and products that we use. And lambda-cyhalothrin and bifenthrin were consistently good for all treatments, but carbaryl, pyrifloxacin, and Pernoff didn't make any difference compared to the untreated. A very minor difference we found with Pernoff with larvae in this very specific instance, but we don't think that that collectively is significant. And throwing this here because something that Matt did and was to look into what happened to those areas the year after treatment. Because if you remember, when we did the end of August treatment, our idea was to target those individuals that would lay eggs and will be responsible for the larva population next year. And that larva population most likely will molt into nymphs and start after overwintering in the following season. So when we look into adults and nymphs, April and June, in areas that were treated by these different products, we saw that lambda cytolytin and bifenthrin, we saw a lower number of adults and nymphs early in the spring, but that tend to disappear and homogenize later than in June. We still saw an effect with the lambda cytolytin that we didn't see anymore with bifenthrin, and we didn't see an effect with nymphs population anymore, but we saw still an effect with the adult population in June. So it seems that the treatment not only control ticks that year, but it might have an impact on the following season. And he did the sticky traps and pitfalls to look into the different how it affects non-targeted species, and here's basically kind of this nice graph showing how the untreated area compared to other areas, and basically this is telling you how different the untreated areas is from the treated areas in terms of pitfall traps, right? Pyriproxen, Pirnoff, and Carvalho were kind of similar to the untreated area, so we collect the same type of arthropods. Lambda cytolytin and bifenthrin that actually had an effect on ticks also had a bigger effect on soil arthropods. Carvalho also had a bigger effect than Pirnoff and Pyriproxen when compared to the untreated. And these are basically some of them are I don't know if I have this here. So you can see the different taxa and how they were affected by different treatments. And Colemba and isopods were probably two of the major taxa affected by these treatments. Okay. And this is with the sticky traps. The sticky traps had lower differences compared with the pitfall traps, and lambda cytolytin in this case was the one that had a bigger effect. This is not a surprise because here it's not only the type of acaricide that you are using, it's also the formulation, right? So the lambda cytolytin was given as a spray, so that's probably why it affected flying insects more than granules. And the conclusions for this study is that pyrethroids were the only effective treatment for controlling ALT. I think this is important because although in our preliminary data, our preliminary study with acaricides, so carbamides also seems to be promising in the field. They didn't do anything, or they didn't do much. Granular bifenthrin was similar to lambda cytolytin. Granular carbamide was not effective against ALT. Growth regulators were not, didn't have a consistent impact on ticks. And granular products might have a greater impact on soil arthropods than foliar treatments. And with that, I just want to spend 30 seconds saying thank you to Julia Gonzalez and to Matt Bickerton that have done the bulk, well, everything that I just presented today. And they have worked extremely hard to produce the data. Jennifer Mora, who I didn't present any of her data, she's working on the in vitro systems. And then I have also a student now, Saria, from microbiology, working on Borrelia burgdorferi, actually working on the role of fatty acids to membrane organization that I hopefully will present at some point later in another seminar here. So with that, I'll take any questions that you have. Yes.
So you said that the ALT is more good at dry climate and they also cluster more. Do you think those two are related? The fact that they cluster and they're
No, I don't think they are related. Clustering we don't know why they cluster. It might be just because they are so many that they just happen to be found together.
So it's not like penguins trying to
No.
cluster.
No, and in terms of there are ticks that are well adapted to drier climates. Species of Ayaloma are very well adapted. Ambioma americana actually is a tick that is very well adapted to drier climates compared to Scapularis. So the adaptation to drier climates doesn't have to do anything with the clustering behavior that we saw. We don't even see, I wouldn't even call it clustering behavior. I would say we see clustering. I don't think that is a tick behavior. I don't think they are doing it on purpose. I think it's just a byproduct of high density. Yes, sir.
So, Alvaro, you have high populations of the ticks in the ecozone, and then you also, when Matt did his experiments, from my understanding, he did a broadcast treatment. Could you just target the ecotone of areas to, reduce the amount of material that you apply, but also provide control?
Yes, I mean, I think when we talk about control, I think it's important to control ticks where it matters. I don't think controlling tick in the middle of the forest make any difference for anyone. If you want to control ticks in the ecotone because people are more exposed to those ticks than to ticks that are in the field. So that will be a way to use less, but at the same time, probably for control approaches to be used consistently, you want to do something that has a lower impact to the environment. So the state's target approach was thought to, in that sense, like to reduce the number of applications. And now Matt has also generated some data on Bovaria bassiana. There are not good studies on Bovaria or Metarhizion on ticks in the field. These studies are typically complicated because population of ticks are heterogeneously distributed. You need to sample a lot to have consistent data and win a good study. Now, with Amaphysalis rangicornis, that changed a little bit because they have the density is so insane that now doing field studies is can give an answer that before was hard to get, and we are planning on doing this type of studies with Metarhizion or Bovaria bassiana that we know some strains work well in the lab, in the field, to avoid the issue of applying acaricides to the environment. Yes.
Was this resistant or susceptibility work done with your H3 colony?
Yes.
Okay. Do you have any colonies for other haplotypes?
No. Actually, we weren't completely sure that my colony was only one haplotype, so we ran it. Andrea ran that when we did the baseline susceptibility study, and all of them were the same haplotype, because we weren't like, well, maybe we just inadvertently put one here and we have different haplotypes, but that's not the case.
I just wanted to throw it out there. There's a paper about this one place where we have some evidence that each one might be linked to, and the ticks there, they did find pyrethroid resistance to. It was H1. So it might be interesting to run on different haplotypes to see.
I mean, they could behave different. Well, I mean, every single tick, in principle, could behave different because they are isolated. They are not reproductively connected to each other. So each clone could be different. So our is H1. Matt, do you remember?
Okay.
Our haplotype in the H3. Okay, so yeah, they might be different.
Yeah, yeah,
Yeah, I think you guys have
yeah. You have H3 global.
Yeah, so that might change with haplotypes. That's true.
You should know already. So these are clonal, and so far we've only detected 3 haplotypes in New Jersey. Although Zoe has new data on that, but so far as we know, there's these 3 clonal lines.
So does that mean there's 3 introductions?
Right. Well, 3 separate female.
Yeah. We don't know if they were introduction at the same time, different instances, two of them together, we don't know. But yeah, 3 ticks at least. Yes.
So you compare the different chemicals, but some in spray formulation, one is in granule.
Yes.
Is that no efficacy because the formulation instead of the chemical?
No, because we have granular formulation for 2 different products, for pyrethroids and carbamates, and pyrethroids work well.
Both in granular formulation?
In granular formulation, pyrethroids work well. Yeah. So it wasn't the formulation; it's that.
Also, you show that CR9 or CR3 has some efficacy and mortality to the ticks.
Yeah.
That means it's maybe sensitive. I know there are some essential oil-based tick control products. Would you think that those would be also effective for the KLT?
I mean, to some extent, they might be. To some extent, they might be. And I don't know if they will be good enough for controlling in the field. Because when you're doing in vitro experiments, well, having positive data is good, but that doesn't really mean that you can transfer that directly to the field. Particularly with oils, I think chemicals are more but I think with oils you will have to do a field study to
Yeah,
prove it.
actually, I had a question on that, on the effect. So I don't remember the scale, but I mean, you had you could see there's increased mortality, but it was really slow.
Yes, it was kind of rapid in the first 7 days, and then it stays and little by little went down. So the first 7 days is the one that we saw the bigger drop. And we just did this. I mean, you have to do this a little bit better, like probably expose it at different times, because we only expose them on filter paper that was treated for 30 minutes. There are different. When you do this with acaricides or with other products, you typically dip. When you are using adults, you dip them completely in the solution, and then you take them out. So we should do it in a more comparable way, because this was just to see if actually mortality was real or it was or not. But now, if you want to compare it with other products and other things, we have to do it in a way that is comparable.
Yeah, but on the other hand, I mean, you use the CR3 in ethanol, right? Using ethanol as the dilution.
Yes.
So the ethanol is going to erode some of the waxes, and then some of the. So that could also be affecting mortality, because it's a dehydrating. I know you're putting them after you dry it up, right? So
Yes, we let the ethanol dry out, and we put them in contact with the paper, but
which is better, because if you dip them, then you're putting them in ethanol, and that's not going to be good. So in a way, what you did was more in terms of checking the effect of the CR3.
Yeah, true. But then, if you want to compare it with what other people did, or with more standard methodology, then you have to use either the larval package assay or the dipping assay, which is what people use in vitro assay.
Yeah, but I mean, when Grayson was doing some of the same kind of thing on mosquitoes, the fact that the ethanol was having this dehydrating effect was something that he had to come up with more of a similar system to what you did, where the mosquitoes are sort of walking on this little treated paper, because that's a better way to look at the effect of the repellent and not the carrier.
Yeah.
So it's really interesting how something that is technically affecting their ability to smell could be having an effect on mortality. Somehow it's messing up their.
Yeah, I don't know. We don't know how that happens, and as I say, the bulk of repellency is due through Haller's organ, but not 100% of it. There are probably receptors in the palps and maybe in other parts of their body that are also accounting for the overall repellence.
Yeah, and the difference between Ixodes and hemapazole is really interesting.
Yeah, that was unexpected actually.
Yeah.
We didn't expect to see big difference between 2 tick species, but yeah.
Right, we're good. So it's 12 o'clock. No, sorry.
Because I'm going to forget about it. We have a Alvaro transcriptome now for ALT. It's
Sounds good. Yes.
Right. Yeah. Right. Well, thank you. Right.
Thank you. So, actually, I didn't check if there were.
Question from Zoom, maybe.
Yeah.
Alvaro, don't forget, we have to go next door for the.
Okay. Yes.
If there's any questions, yeah.
I don't see any.
Solved now?
No, it's because I had the presented mode before I just. Yeah, I don't see any questions here.
That was your
Now I think we're
schedule.
good.
Okay.
So what are you? Are you like a student, right?
I'm just
Okay. Bye, everyone.